Alrighty.
Hey, so cool. Welcome. This is Emmy Moore Power's 7th grade science class. We get to participate in a cool demo with Williams College. It's what are we looking at today?
Scanning Electron Microscope.
Electron Microscope, yeah. So I'm going to turn up the volume here. I'm going to see if I can get the speakers going too, but hold on. One last thing after all this scrambling, and I want to make sure that I get you some good sound here. Someone took my cable. Okay, so we're going to have to go with this little speaker, but I think it will work okay. So you guys will have to be kind of quiet, but you'll listen, and then Nancy will. How do you want to be called? How should
Nancy's good.
Nancy's great. So
Yeah.
She's there live in the studio. You can see her up here on the screen, and we're broadcasting. You can see some of the apparatus back up here. And why don't we just listen to her talk a little bit about what she does and how she runs the equipment, and then maybe you guys can think of some good questions while you're in this process. If you have some ideas, write them down so you can pose that question later at the end of the session. Okay? Cool. Thanks.
Well, have some of you already seen this in 5th grade? Did you come with Miss Maselli and use the electron microscope? No?
Yeah.
Maybe one person? Yeah, this is the electron microscope, and we use it to look at cells. And I'm the lab tech for the scope, so I help the students get their samples ready and run the scopes. And I have some plants in there, so I'm going to, the beam is on. We have an electron beam. So I'm going to see if you can see what I'm trying to look at. Let's make it bigger. Can anybody guess what it is? I can bring this closer if that helps. Is it too wiggly?
That's better.
It's better?
Yes.
Anybody want to guess? It's not an animal. And can you guess what I'm doing when I go like this? I'm magnifying it. The round things? Anybody want to guess what the round things are? Well, they are, this is a stem of geranium, and I've simply sliced it so you can see the inside. And the round things are just the cells. So if you magnify it enough, we're at 1,200 times, and the little round things there are probably chloroplasts. Don't ask me where the nuclei are, not in the plane of section. So I will move that back just to make you really dizzy. So that's the stem of a... Uh-oh! I didn't see anything. Do you see anything? Oh, it's just going really slow. So,
Nancy, did you say what they were looking at there?
yeah, it's the stem of a geranium, and I've sliced it.
Okay.
Now, is anybody good at the whole monocot, dicot thing? These are the vascular bundles.
Okay, we have not gotten there yet. We'll get there in the spring with looking at monocots versus dicots. Types of flowers.
I think it's a monocot. So here, I'll focus it for you. And see, it's got like, whoopsie. You can change the speeds on it, and that makes it easier to focus. And it's got like bed springs coming out of it. Can you see the bed springs? So here, I'll slow it down for you. So those are the plant cells, and those are the vessels that make the water go up and down the plant. And these secondary wall thickenings come out of it. So I'll show you the leaf. Because that's the stem. And I'll show you the leaf, because it's over here. So you can move things around. Can you see my stage? And we're inside of a vacuum. Anybody want to guess an advantage of this scope?
It's a scanning electron microscope.
So you can see stuff in 3D.
Yeah, 3D, exactly, exactly.
So you're showing us a leaf right now, correct?
I am. Can you see it okay?
Yes.
Yeah. Oh, okay.
Structures you never think about, right? You can see like they look like little hairs coming out. That's probably the fuzz.
They do. They do. And you are looking at a leaf, and it is 525 times magnified. Can you think of a disadvantage of this microscope?
You get to see more.
She asked for a disadvantage.
You can't see the whole thing.
Okay. So Julia, talk louder.
You can't see the whole entire thing at the same time.
Yep, exactly. You can't see the whole thing at the same time. Yeah, as you can tell, I've actually cut up this leaf and I've chopped the stem in half. So yeah, our field of view is small and you can't see the whole thing at the same time.
And Talia also said you can. This is in black and white. It's not in color.
Yeah, exactly. It's in black and white. You may see. I'm going to grab something here. People put scanning electron microscope pictures in magazines, and they always add color, and you shouldn't believe it. It's all fake. Can you see that?
Yep.
Yeah. That was in Time magazine, and it's pink, but it was from a scanning electron microscope, so it was originally black and white. And it's a white blood cell. I don't know if you can see the tiny blue dots on the top, but that's the AIDS virus on a white blood cell. So, and they've colorized it, and it's very pretty in a sense in pink and blue. So when you see these pictures in color, you know they've been faked. So we can easily see viruses or cells or a bacteria with this microscope. I'll switch and I'll put on the bacteria for you. So because they're all a vacuum, usually things are fairly dead. So that's the other disadvantage. If you want to look at live cells, you want probably a light microscope. So I'm going to twirl the stage. I don't know if we're going to be able to see it. Can you see the stage moving?
Yep.
Okay. Let's see. I've got to turn the contrast up. Well, this is the next one. It's not the bacteria, but they're small. So here, I'll have you guess. They kind of look like little spaceships.
Bacteria are very, very simple cells. They have a cell wall. They have no nuclei. Okay.
Yeah, I didn't quite get to the bacteria. I just went to the next one. Does anybody have allergies?
Yep.
Can you guess what these are? Oh, somebody said it, I think.
They're being shy.
I think I heard somebody say it.
Pollen.
Yeah, pollen. Somebody's got it. That's from, whoopsie, milkwort. That's a little teeny plant up at the Clark. So here, I'll try to find the bacteria for you. I'm just making the magnification go down. And here you can see the stage swirl. That's a blank. Oh, that's a blank. Well, no, there's something there, but it's big. I'll go back to that. This has got a label. That might help. Yeah.
Looks like
So I don't know. Whoa. You probably can't see it from there, but the image is like really squiggly. So I'm trying to get it so it's not squiggly. Oh, so now it's not squiggly. And there's one. And those are the bacteria. This is B. subtilis, and it's similar to E. coli. It's a rod-shaped bacteria.
E. coli is the one that's in the news all the time that makes people sick when it gets like in meat and stuff.
Yeah, that's the one.
Yeah.
Yeah, yeah. Although the good E. coli in the gut are a nice thing, but the bad ones in meat are not so good. And the size bars here. So this is 20 micrometers. So the width of a bacteria, these bacteria is about 1 micrometer.
1 micrometer?
Yeah. Which is smaller than a millimeter.
Yep, yep. I even got my chart out, so there are, so I wouldn't forget, but there's 1,000 micrometers in a millimeter. Oh, here, I'll switch samples. And the samples are all on little holders. And they look like that. So whoever said you can't see the whole thing, they were right. You can't see the whole thing. So I'll switch it one more time.
You could tell them a little bit about the preparation of the specimens.
Oh, sure. The bacteria, they're kind of soft and squishy, so the sample prep is a lot longer than, say, bugs. You have to fix them in glutaraldehyde and dehydrate them to get the water out, and then coat them with a gold palladium to make them conductive. If you don't do that and you just put the bacteria in the vacuum, can you guess what they would do if you put soft cells in a vacuum?
They would get crushed.
I'm sorry?
They would get crushed.
They'd get crushed, exactly, yeah. So we go through a long process so that they don't get crushed.
that's
Which again, you know, if you want live cells, you don't want the scope. So I don't know if you got to see the video of the liverworts. The preparation for that is similar to for the bacteria. And I will switch the sample for you here.
So I'm going to show you the, I have a little video clip that she did with a student actually showing a microtome, which is a little teeny tiny knife that was cutting part of a very small plant so that he could make a microscope slide with that. I'm going to show that to you when we get my computer hooked back up.
Oh, I'm glad you got that.
Yeah.
Yeah. We had a student volunteer for that. We kind of coerced him into it. He was really good about doing it. Anybody want to guess what this is? You use them like all the time.
She's asking you what you guys think this might be. Can you bring the camera a little bit closer?
I can.
bring the camera closer.
Oh, I know what it is.
You know what it is? Yeah, Richard knew right away, too.
Yeah, Sierra just got it.
Yeah, it's a like a
Microchip, yeah, or I don't know.
Awesome.
Yeah.
Yeah,
There's a
that's
hole in
so
it. It does look like
Imagine soldering that by hand.
Yeah.
Yeah, I know. It must be very.
So I don't know much about them, but the computer science people tell me these little things are transistors. Does that sound right, Richard? I don't know. But anyway, if you go down, it looks like a little cityscape.
Yeah.
There you go. I'm cleaning it up. Of course, if I didn't wiggle the camera, it'd be good.
So,
Yeah. So microchips are easy because they're like dry, and you just coat them with gold, make them conductive, and throw them in the scope. Unlike the bacteria, where you have to go through a process to get them ready. Anybody got any questions?
Go ahead, Julia.
do you ever have you ever thought of like making microchips while using a microscope? So,
You know, I think they kind of do that with it's similar to this, because as you can see, we have nice high mag on this, but we have an electron beam on this scope, and I think the gadgets they use to make chips are called focused ion beam microscopes, or FIBs, and they are used to, now Richard might know more, Amy, you might know more, they're used to pattern the surface, and they're computer controlled, and they're used to etch the surface. And it is an ion beam instead of an electron beam, if I understand it correctly. So you're absolutely right. There is a similar gadget to this, except for they cost like, I don't know, well over $1 million, those ones that they use for the microchips, and this was nowhere near that. So, yeah, exactly.
Maddie has a question. Maddie?
What's the largest magnification the microscope can go?
Oh, well, that's always fun. So we can do that, or we can try to do that. So we're at 400 times now. It's pretty low. You could do that with a light microscope.
Yep,
So now
that's what
we're
ours.
at 1,600. So you couldn't do that with a light microscope. So that's a good thing. I'm going to keep going up in mag. Now we're at 6,000. Let's face it, it doesn't look great, but it's, I mean, you can half see it. 6,000. So how high does it go? Let's just keep trying it. 12,000. Let's face it, it's getting a little blurry. 24,000. Blurrier yet. So eventually, this is 50,000. So,
How much?
and that's 100,000. And what we've got here is what I call empty magnification. You're magnifying it, but you can't resolve your image. So I would say the useful mag is, what do you think, Amy, the useful mag is? 13,000 is good.
Yeah.
Yeah. 50,000, not so good. 100,000, we can't really do it.
The stuff I've read goes between 12 and 15,000 for this kind of a microscope.
Yeah. That's about it.
Can I ask a question?
Yeah.
So, if you're looking for something that's at that cellular level, that's a virus affecting a blood cell, for instance, well, how do you, when you're prepping the specimen to look at, how do you even know where to find that? And
It's a problem. You're absolutely right. It is a problem.
Needle in a haystack.
Yeah. I briefly worked in a diagnostic lab, and they thought this poor man had encephalitis, and they were trying to find the viral particles in a tiny tissue sample, and they never did. So it's a problem. When you're studying the cells, you grow them up on a cell culture, and you intentionally infect them with the virus and grow them at the ideal conditions so you get lots and you can see them. But in their native state, it's a needle in the haystack. So yeah. I see a hand.
Alex? What's the chemical process to put put the bacteria in the slide, and then like how long would that take?
Yeah. The bacteria for these, we did fix it, dehydrate, and put on the sticky tape and put in the microscope, and that takes a minimum of about 4 hours. So, and if we're using the other microscope, it takes a couple days. So, but yeah, a couple hours. So not quite quick at all. The microchip, you can throw it in and look at it because it's dry. But cells have so much water in them, and we're using a vacuum that you have to do the process. Yep, a couple hours.
Lucy.
Do you ever need to use the 50,000 magnification?
Oh, well, we have the students in the chemistry labs make their own nanoparticles, which are very small particles made out of, they've been doing them out of nickel, sometimes other substances, and we can barely see them on this microscope, so we would like a bit more magnification for the nanoparticles. That's a real hot field in nanotechnology. But yeah, 50,000, it's a problem.
Because the higher in magnification you go, the lower the resolution.
Yep, exactly.
Fuzzier and fuzzier. I had a question. These guys spent part of last week looking at cells underneath a light microscope, trying to find organelles.
Oh yeah. So you would want our other microscope for that, but that is not 3D and it's hard to look at. But organelles, if you're using the other microscope, the transmission electron microscope, you get cells that look like that. That's one cell with the nucleus and the nucleolus. There's the nucleus. And here's the ribosomes and the cytoplasm. So this is part of a chloroplast from a plant. This is the cell wall. That's the chloroplast. So this is not 3D, it's 2D, and it's the other microscope. But that one's hard to look at. You have to sit in a dark room with green screen. Oh, white blood cells again look like this. They're white blood cells. And the students took this picture right here at the college.
Oh, very cool.
There's the nucleus, cytoplasm. And if you crank up the mag enough, you can see Golgi and mitochondria. Trying to see what I've got here. Yep. These are nerve cells.
Yeah.
So you know, you can use all three microscopes. You can do the light microscope, you can do this scanning electron microscope, or the TEM. The TEM, you can see the insides of cells, and this one you get the 3D and you see the outsides of cells.
Julian, have a question?
Yeah. What's your favorite field of science?
What's my favorite what in science?
Field of science. Like what?
Field?
Yeah.
I like the plants. I do. I think that's my favorite field. Yeah. Although the material science stuff is fun too. And the material science stuff, you know, the way you can magnify this, it's very good for forensics. If you're doing fiber analysis at a crime scene, or you have small particles and you want to know what they are. If you have particles, we have a gadget on this that tells you the chemistry. So you can analyze a piece of dust at a crime scene. Very handy. But I think my favorite is the plants.
Can I ask another question, quick one?
Oh yeah, sure.
So if you had the 3D capability, can you also gather 3D data that we could use to, because we have a 3D printer here. Is it?
Oh, You know, a lot of people are asking me that lately, and the answer is no. We only have, it looks 3D, and we have the X and the Y information, but we don't have we can't measure the actual height unless we tilt it on an edge. So to get real 3D information, you want the fourth microscope we have here, the atomic force microscope, that gives you true 3D information in all three dimensions. And you get something that looks like this.
Yes.
Yeah, and that's actually atoms of carbon. So
Really?
with that microscope, you can see atoms, and you can also look at, the problem is it has to be really small structure, but you do get the actual 3D, and you can give those coordinates to a printer and make an object. But unfortunately, not with this microscope.
Soon. Hopefully, soon.
Yeah, hopefully soon.
Yeah, we printed the comet. We got the 3D data from the European Space Agency, and we printed a comet.
Oh, very cool.
Yeah. So.
Have you got your own printer there?
Yeah, we have a TAS4, just like Williams, the Bronfman Center, and also your makerspace at Sawyer.
Oh, that's very cool.
Yeah. Cool. Any other questions? Well, thank you very much. Yeah.
You're welcome.
This was really great, and now we have a Google Hangout on air for us to kind of listen to and review and stuff like that. And it was really great to see everything here in the lab. Fascinating work.
It's good to talk to you.
Thank you.
And I hope the rest of your day is great.
All right. Bye-bye.
Bye.